Good morning and good evening, everyone. I would like to welcome you to today's Cyagen webinar. My name is Qiming Wang. I'm the Marketing Director here at Cyagen. We'll talk about an extremely popular topic today, that is GLP-1 and its related drug in body weight management. Alongside me is our guest speaker, Dr. Anna Wu. Dr. Wu is the Study Director at Cyagen. Dr. Wu brings exceptional expertise in metabolic disease research. Dr. Wu earned her PhD from the University of Groningen, where she invested therapeutic intervention for metabolic dysfunction-associated steatotic liver disease, MASLD. Currently, Dr. Wu is leading Cyagen's metabolic platform, where she directs preclinical studies leveraging her specialized knowledge in vivo pharmacology, translational animal models, and therapeutic development for obesity, diabetes, and hepatic disorders. Please feel free to type in your questions in the chat during Anna's presentation. We'll answer as many questions as we can. Anna, so since both of you and I were at the ADA meeting back in Chicago, can you please share us some key takeaways from the meeting?
Okay, thank you for inviting me to present at today's webinar. And I'm the study director for the Metabolic Disease Efficacy Study platform at Cyagen. And I'm honored to share our services and insights into anti-obesity research. Early this June, Cyagen also participated in the American Diabetes Association 2025 conference in Chicago, where we were excited to hear about the latest progress in clinical trials for anti-obesity drugs.
Great. In her presentation today, Dr. Wu will address a critical gap in obesity therapeutics, such as GLP-1 or agonist drugs, that is muscle wasting and metabolic adaptation. So without further ado, I will pass the stage to Dr. Wu. Anna.
Yeah, thank you. Then I'm going to share my screen and to present. The topic I would like to share with you is slimming science, harnessing the power of GLP-1 and Amylin for obesity care with reliable preclinical in vivo models. Here is a quick look at today's agenda. We will begin with some background, setting the stage by looking at current obesity trends and the key therapeutic targets. Next, I will introduce our services, especially how we utilize validated animal models to support pharmacodynamic evaluation of GLP-1 and amylin-based therapies. And finally, we will conclude with a Q&A session, where I will be happy to take your questions and hear your thoughts. Obesity is defined as having a body mass index, or BMI, over 30 kilograms per square meter. It is a chronic disease characterized by the excessive accumulation of body fat. Obesity significantly increases the risk of developing conditions such as type 2 diabetes and hypertension, and it is associated with a shortened life expectancy. According to global forecasts, obesity is expected to affect 25% of the world's population by 2035. Research has consistently shown that obesity raises the risk of many serious health problems, ranging from heart disease and diabetes to liver disease, sleep apnea, depression, and even cancers like breast and colorectal cancer. Obesity is a chronic disease. It's characterized by excessive or abnormal distributed adipose tissue accumulation. There is a part of pathophysiology of obesity is the imbalance of energy homeostasis and metabolic adaptation. The three primary components of energy homeostasis are energy intake, expenditure, and storage. Obesity arises from distributed energy homeostasis characterized by excessive TG storage in white adipose tissue and impaired thermogenic capacity of brown adipose tissue and benign adipose tissue. Mitochondrial dysfunction and suppressed white adipose tissue browning perpetrated metabolic inflexibility, driving insulin resistance and systemic metabolic disease. Interventions targeting adipose plasticity and mitochondrial health hold therapeutic potential. So far, the most effective interventions for obesity include lifestyle modification, weight loss surgery, and pharmacological treatments. Among these, GLP-1 based drugs have recently gained the most attention due to their strong efficacy in promoting weight loss. However, researchers are still actively exploring new anti-obesity drugs, while also working on strategies to minimize side effects and improve long-term safety. We now know that GLP-1 receptor agonists are highly effective for weight loss. This has sparked the rapid development of newer drugs that combine GLP-1 activity with other hormone pathways to boost effectiveness, not just for weight loss, but for broader metabolic improvements. These include dual agonists like GLP-1 and GLP, and even triple agonists that also target the glucagon receptor. Let's quickly go over how each of these hormone works. GLP-1 receptor agonists help with weight and blood sugar control in multiple ways. They act on brain, especially the hypothalamus and brainstem, to reduce appetite. They also slow down gastric emptying, increase insulin release when blood sugar is high, suppress glucagon, and help preserve or increase the number of insulin-producing beta cells. GLP receptor agonists also help stimulate insulin secretion in response to glucose, although this effect is often reduced in people with type 2 diabetes. GLP can also raise glucagon levels, promote beta cell growth, supporting bone formation while reducing bone breakdown, and improve blood flow to fight tissue after meals. Glucagon receptor agonists, on the other hand, mainly act on the liver. They raise blood sugar by promoting the breakdown of storage glucagon and stimulating glucose production. By combining these different pathways, dual and triple agonist drug aim to create more powerful and longer lasting effects for managing obesity and metabolic disease. Recently, most researchers have started shifting their focus towards increasing energy expenditure as a strategy for weight loss. One promising target is amylin, a hormone that's released along with insulin. Amylin plays an important role in regulating blood sugar after meals. It helps by suppressing glucagon, slowing gastric emptying, increasing satiety, and possibly even boosting energy expenditure. On top of that, amylin may also offer cardiovascular and neuroprotective benefits. Amylin functions as a synthetic hormone released into the bloodstream by beta pancreatic cells. Amylin activates various homeostatic and reverse centers in the brain to suppress appetite and reduce food intake. In summary, obesity is becoming a serious global problem. It affects over 1 billion people worldwide and brings huge health and economic burdens. GLP-1 based therapies have really moved the field forward, showing meaningful weight loss results, but the challenge is that all current FDA-approved treatments eventually hit plateau, which limits their long-term effectiveness. That's why we urgently need next generation anti-obesity drug, ones that can overcome this plateau and target multiple metabolic pathways for better sustained results. At Cyagen, we have established several animal models for metabolic disease, including obesity, diabetes, MASLD, MASH, and atherosclerosis for preclinical drug testing. In addition, using Cyagen biomodel resources, we have conducted many humanized animal studies. For example, we have worked on inhibin and GLP-1 related models, which are gaining popularity among biotech and big pharma companies developing siRNA therapies. Our facility is fully equipped with tools for daily monitoring, muscle function testing, metabolic cages, body composition analysis, and biochemical assays. In this study, the mice were induced using a 60% high-fat diet from research diets, and the mice were induced for 20 weeks to establish obesity. We treated them for 4 weeks with different drugs: Tirzepatide, a dual GLP-1 GLP agonist; Retatrutide, a triple GLP GLP GCGR agonist; and MASLD, a dual GLP GCGR agonist. Each at low and high doses. The drugs were administered subcutaneously every 3 days. The drugs were administered subcutaneously every 3 days, as expected from the mechanism of GLP-1 agonist. We saw a rapid drop in food intake after each dose, leading to body weight loss. Among the groups, Retatrutide showed the most sustained effect. The mice continued to lose weight steadily, while the other groups at around 20% weight reduction. We also monitored fat and lean mass changes by day 7, day 14, and day 28, respectively. The reduction in fat mass was clearly significant. Interestingly, only the high-dose Retatrutide group showed a statistically significant reduction in lean mass, indicating a potential side effect at that high level. This slide shows the blood chemistry from the drugs can prevent liver function levels by high-fat diet-induced ALT and AST levels. also had effect on TG, LDL-C, and HDL-C levels. As you know, GLP-1 drugs are widely used for diabetes because they effectively lower blood glucose and improve insulin sensitivity. In our study, we measure fasting blood glucose every 2 weeks to monitor the effects. After treatments, all the drugs were able to reduce blood glucose level, dominantly showing clearly efficacy in all groups. After treatment, we collected adipose tissue, small muscles, and liver samples, and measured the weight of each tissue precisely. We found that all the drugs reduced the fatty liver weight and decreased the white adipose tissue. Interestingly, retatrutide also caused a significantly reduction in soleus muscle mass, which aligns with the overall loss of lean body mass we observed. Our obesity model was induced by a high-fat diet for over 20 weeks, and at the time point, the liver showed features similar to MASH. All the drugs prevented steatosis and ballooning in the liver, but they didn't reduce inflammation. Overall, GLP-1 drugs showed good efficacy against fatty liver disease. There is a growing research on the crosstalk between adipose tissue and the liver, especially regarding their role in inducing skeletal muscle hypertrophy and reducing fat mass. To evaluate this, we quantified the adipose size using HE staining of perirenal white adipose tissue. We observed that retatrutide significantly reduced the adipose size by more than 40% compared with the DIO model group, and also increased adipose density. This clearly shows that retatrutide has a strong beneficial effect on white adipose tissue in mice. This slide shows we performed the muscle fiber cross-sectional area was measured from HE stained section of muscles. The difference between the retatrutide group and the DIO model group was not statistically significant. This slide, you will see that we can also assess muscle function using grid hanging test, although this study didn't focus 100% on muscle enhancements. These results provide valuable insights. With high-dose retatrutide, the mice showed much better performance in the grid hand test compared with other groups. For the analysis, we use one-way ANOVA, taking the DIO model group as the reference. At Cyagen, we provide well-validated and highly reproducible DIO models that can be used for both monotherapy and combination therapy studies. Our protocols cover a wide range of endpoints, including body weight, glucose metabolism, hormone levels, and histology. Throughout the study, you will receive regular weekly updates, and at the end, a complete study reports. We also offer flexible support, such as custom dosing regimens, positive control groups, and even mechanism exploration. These features make our perform ideal not only for enabling studies, but also for early stage drug screening. As a final note, I would like to emphasize that at Cyagen, we are very open to cooperation opportunities, including GLP-1 and amylin drug screening, obesity-related animal models, and customized pharmacodynamic evaluations. If you are interested in technical discussion, project cooperation, or potential partnership in the preclinical evaluation of anti-obesity therapies, we would like to we are happy to connect with you. And thank you very much for your attention. We sincerely look forward to working with you.
Thank you, Anna, for sharing the comprehensive preclinical study with GLP-1 agonists, dual and triple agonists, as well as demonstrating our CRO capability and the differentiated animal models. Just for audience information, we have the GLP-1 receptor humanized mice, as well as the GCGR and the GIPR humanized mice. If your candidate drug is not cross-reactive with mouse, you may also consider those humanized mice. So, Anna, in your presentation, you mainly discussed the DIO mice, but I do find some researchers like to use DIO rats. So, one question from the audience is that when should people consider rats for their study, and when people should use mice?
Yeah, thank you for the question. DIO rats and DIO mice each have their advantages depending on the goals of your study. DIO rats are typically preferred when you needed a large blood volume or repeated for sampling, you need more tissue for biochemical or histology analysis. So they also tend to develop more pronounced metabolic features such as insulin resistance and dyslipidemia, which are useful for mimicking human metabolic syndrome. And now the research on amylin target, most studies are conducted in DIO rats because this target showed weaker activity in DIO mice. So at this moment, we are establishing DIO rat models now, and we will share the full pharmacodynamic data on amylin targets in these rats in our upcoming work. Thank you.
Okay, great. And the next question is, how can we determine whether a drug's effect is limited to reducing food intake, or it also influences other aspects of energy metabolism? So how should such an experiment be designed and carried out?
Yeah, thank you for the questions. And this question is also very, the clients considered a lot recently. So to distinguish whether a drug only affects food intake or also learns energy metabolism, a pair feeding experiment is commonly used. In this design, a control group is fed the same amount of food as the positive drug group, matched for caloric intake, but without receiving the drug. And if the drug treatment group shows a greater weight loss or metabolic changes compared to the pair fed group, it suggests the drug has additional effects beyond appetite suppression, such as increased energy expenditure or fat oxidation or other functions. So, yeah.
I wish people also can have controlled food intake. All right. And so the last question is, what are the biggest challenge with GLP-1 and amylin drugs today? Especially given your recent participation in ADA, what are some like future directions?
Yes, from the ADA conference, we can see many clinical trials. So the main issues are the tolerability of gastrointestinal side effects such as vomiting and diarrhea. But the biggest concern now is that weight loss often comes with muscle loss, so which could affect long-term health. The future drug development may also focus on the co-therapies that help preserve the muscle mass. Yeah.
Okay, I think that's all for today's webinar. And Anna, thank you again for joining us today. And I just want to thank the audience again for watching the webinar. And see you in the next episode. Bye-bye.
Okay, thank you. Bye-bye.